Hello, Eastview Scholars. Welcome back to Sharp Shorts. Here, we're going to talk about the phases of mitosis. We just went through the cell cycle in one of our last videos, and the question is, how is this that we happen to go through this wonderfully colored phases of mitosis, or what are these phases that cause a single cell to become two? And again, really the key to this is that particularly with multicellular organisms, that you keep these linear strands of DNA, keep their integrity so each cell gets the right complement. It's a bit easier with prokaryotes and binary fission, which we'll talk about after this. Anyhow, here's the cell division. Here's the phases of mitosis in order. Prophase, prometaphase, metaphase, anaphase, really nice girl, and telophase. Cytokinesis kind of overlaps the telophase, and again, some people include that in telophase. The idea, though, is that while these phases are dividing the DNA appropriately, the cytokinesis divides the cytoplasm in organisms. And that's not exact, not organisms, but organelles. That's not exact as it is with the DNA. But it's just dividing the cytoplasm and the stuff in it. Basically, here you go. Here we were in interphase, and if you remember, that's 90% of the cell cycle. And as we get close to prophase, what you start seeing, and this is for animals, you get a couple of centrosomes. Plants don't have centrosomes. It's one of the things that distinguishes animal cells. Even though plants go through mitosis in the same phases very similarly, they don't have those centrioles to help with the movement. Anyhow, interphase, as you get close to that mitotic phase, is where we're at G2. When you hit prophase, here's the key. You've had these centrosomes, and they're going to start migrating towards the other poles. And while they're doing that, they're forming microtubules. Now, how is that occurring? Well, they break down some of the proteins of the cytoskeleton to use here. The nuclear envelope is not quite dissolved here. By prometaphase it is. But what you can see really in the metaphase, prometaphase, the chromosomes have condensed. Of course, they were copied in the S phase. You've got this migration and the formation of these microtubules, and you get some that bond right to what are called kinetochore proteins. In the centrosome here, there's a couple of proteins, one on each side, where those microtubules combine, because then it'll allow to pull the sister chromatids apart. The ones that don't attach to chromosomes really use each other to help push and elongate the cell and give it that room to do what it's got to do. And again, these are formed from some of the proteins in the cytoskeleton and stuff that aren't needed during this process. Okay, so that's the key for each. And here it's just the division of the nucleus and the formation of the spindle. And you can see the aster formation of the centrosome there. Then by metaphase, what you have is the alignment of the chromosomes in the middle on what's called the metaphase plate. And really here's a checkpoint too that we'll talk about in cell cycle, is each of these kinetochores has to have an attachment, or you could get a process called non-disjunction. The ones that have not attached to the kinetochore, you notice, are helping elongate the cell. Okay. Once they're all aligned on the cell and all these microtubules are attached to the kinetochores, then you have the anaphase, and that is the shortest of the phases. And that is just where the sister chromatids then are pulled apart by these microtubules and go to each pole. And then the telophase and cytokinesis in an animal cell is the formation of the cleavage furrow, basically pinching off. You see the nuclear envelope coming back together and forming. The chromosomes will then stop being condensed, and they will spread out, and they will do these things. Now, while that's happening, these spindles are broken down. Those proteins will go back into the normal microtubule and cytoskeleton type proteins that they normally do. Here's another picture just showing the metaphase plate and stuff, and I like this micrograph that shows you the microtubules and the binding to the kinetochores. Okay? Now, one of the questions is, as you go through anaphase, what really happens? Is this thing being reeled in, and is there motor proteins here, close to the aster, the spindle pole, the centrosome, that reels these in like a fishing pole, or are the motor proteins really here, and do they walk? If you think of that film we saw at the beginning of the year where you had the little walking guys, do they walk along like they do with vesicles and stuff like that? Is it degraded here? As it pulls it forward, does it degrade after it reels it in, or do the microtubules degrade behind? And work done at the University of Wisconsin, where they selectively tagged for fluorescence the spindle fibers, and then took a laser and cut the mark. What it appears is that the motor proteins are here as a part of the kinetochore, and they walk along the microtubules, and they're broken down to be used in the normal cytoskeleton after. There is some evidence coming out, though, that it's a combo both, that there is a little reeling in kind of fishing pole thing there, but that is a current state of study. Okay, now here you see, because of the cell wall formation mitosis in plant cells. And the difference that we have to think of here is that cell wall. That's the protective and structural formation here. So what you don't see when you get to this cytokinesis is you don't see in the telophase a cleavage furrow and a pinching off as you would in animal cells. But what you see is formation of a cell plate that will then go across and divide the cell in two. Okay? And that's a difference. Besides the centrosome, that's another difference in plants. Okay? As illustrated here. Okay. Now, binary fission, let's talk about that. It's a little simpler. That's mitosis, that's reproduction in single cell. And yes, they go through mitosis. But oftentimes bacteria is a single strand of circular DNA. So while it does need to keep the integrity of the DNA, it doesn't have all the phases necessarily of anaphase and keeping it separate. Really, what you have here is an origin of replication that says, here's where we start, and start copying. And so really it just goes around the circle and spins off a copy of the circular DNA, which then again, you know, one origin on each side kind of migrates after you get this origin and you start copying it. It migrates, and your circles get on each side, and then it divides. Really, so really not a whole lot different, except it is less complex in having to keep single-stranded DNA separate and from fragmenting and catching at each other. Anyhow, that's our basic phases, and you can see how those spindles work and stuff to keep the DNA separated. Our next film will talk about the controls and when does a cell know how to go through this. Until then, Scholar on, Eastfield.